सो हेलो बच्चों, हम जानेंगे मिनरल न्यूट्रिशन. सो व्हाट इज मिनरल न्यूट्रिशन? मिनरल न्यूट्रिशन आर द inorganic compounds or inorganic nutrients that is mostly present in soil and food. These are helping mostly or functioning as animal or plant. So exactly that help in development and growth of the plant and animals. So this mineral nutrition different types. One of the technology first discovered by one of the scientists Julius von Schatz. This technology is known as hydroponic technology. This hydroponic technology, in this technology, through nutrient media, we grown up a new plant without absence of complete absence of soil. This element, different elements that observe plant mostly among 105 elements, plant observes 60 elements. These are nutrients, different type of minerals that observe plant. One is called phosphorus, sulfur, argon, selenium, silicon, calcium, iron, nickel, molybdenum. These are most important nutrient or minerals that helps in growth and development of plant. So first I'm discussing what is the exact criteria for a essentiality or essential nutrient or essential mineral. For the criteria, one is known as that helps in normal growth and reproduction. That helps in normal growth and reproduction. And the second characteristic, that is most specific and non-replaceable. What is non-replaceable and specific? Let's consider, we know nitrogen. If nitrogen helps in building nucleotide, this nucleotide is built by nitrogen. We cannot replace without nitrogen. Means it is the basic composition of nitrogen. So without nitrogen we cannot do these things, or the basic backbone of nucleotide is nitrogen. So as like and specific, let's consider this nitrogen have specific function to build this nucleotide as like or different constitution of protein, and this protein act on different things, so it have a specificity. And second is most be directly involved in metabolism. Most of the nutrients, most of the minerals that directly involve in metabolism. So different type of essential element are divided according to quantity required and their function. So according to quantity required, it is divided into two types. One is known as macro and another is called micro. Macro elements are mostly present in 9. These are calcium, carbon, hydrogen, nitrogen, oxygen, phosphorus, sulfur, magnesium and potassium. As like micro are 8 molecule. These are nickel, zinc, copper, iron, manganese and molybdenum. So these are divided according to micro and macro. Where here you see this macro are 9 and micro are 8. Now I am discussing the essential element according to the function. So according to the function, it is divided into different types. First is known as structural element. Let's consider this is a part of skin in animal. If I am taking this skin that consists of a protein called collagen. This collagen consists of carbon, hydrogen, oxygen, nitrogen elements. So this is the structural element, means this is the most part that constitute or basic backbone like constitute the all the things. Next is enzyme. So we know enzyme that require, this is enzyme that require a cofactor. Why it require a cofactor? It require a cofactor for a proper functioning. One is known as molybdenum. Molybdenum that act as a cofactor of nitrogenase enzyme. This nitrogenase enzyme helps in nitrogen fixation. Next, Zn. Zn means zinc that act as a cofactor of alcohol dehydrogenase, as like magnesium. This magnesium that also help in PEP carboxylase and Rubisco, and that act as also a cofactor of these enzymes. Another concept according to function is known as alter osmotic potential. So what is osmotic potential? Osmotic potential is the potential of water or the potential of water. Means let's consider this is called psi W. We know if the solute increases, then what happen? Water potential decreases. If the solute decreases, the water potential increases. As like the most in stomata, the one factor, one nutrient or one mineral, this is called potassium. This potassium helps in closing and opening of stomata due to solute concentration. If the solute concentration in the cell solute concentration or potassium ion increases, that rise or that helps in rising the water potential, that helps in first decrease the water potential, and due to the difference in water potential, water enter from nearer cell to the this specific cell of stomata, and that helps in the opening of stomata. Next is energy related. So energy related, one of the concept is known as magnesium. This magnesium helps in chlorophyll synthesis and chlorophyll constitution of chlorophyll also. And phosphorus, this phosphorus is the backbone of ATP, adenosine triphosphate. So all are the discussion of quantity according to quantity and function. In function we see structural element, enzyme action, alter osmotic potential, and different type of how energy, different mineral nutrient that helps in energy. Next, another concept that is called toxicity of micronutrient. So what is toxicity of micronutrient? Toxicity means if any mineral at a saturated level, means at a balanced level, let's consider we need X mineral 10 microgram. If that provide or that taken by plant 20 microgram, then that shows toxicity to the plant due to at enter into the cell and creating toxic or create due to that create toxicity in the cell. So this toxicity is caused by the increase in the level of specific minerals or high. This is the increase in minerals. And second, another concept, one is known as the concentration essential for plant growth and development. This concentration is known as critical concentration. Without this concentration level, let's consider it require 5 microgram of nutrient, this specific nutrient. If not this specific nutrient available to the plant, then plant cannot survive or can show deficient of this nutrient. So this point of nutrition is called critical concentration. And another thing, any mineral ion concentration on tissue that reduce the dry weight of tissue by 10%. If the plant decreases 10% of dry weight, then it is called plant intoxic. That plant shows toxicity. One of the mineral, this is called manganese. If this manganese taken more or due to toxicity, or if it is increase in the cell, let's consider this manganese concentration increase in the cell, that shows toxicity. Due to this toxicity, one brown spot found in leaf, and second case is iron, calcium and magnesium that cannot, here is let's consider this is iron, calcium and magnesium that cannot uptake by this cell because manganese is shows high toxicity in cell, so these are not taken by or uptake by the cell. Role of different mineral elements. So these are, one is called nitrogen. First is nitrogen. The nitrogen plant absorb in NO3. What is NO3? That is nitrate, nitrate, and that require, mostly this nitrogen helps in meristematic tissue and metabolic active cell. So why we know this meristematic tissue that are mostly found in growing regions. Let's consider one of the apical part of the plant that also consists of meristematic tissue and a metabolic active cell. During cell division, we know different types of protein, nucleotide are grows DNA also, and this protein, nucleotide, RNA are composed of all the all the component. The basic composition is nitrogen. So we know in nucleotide also we found nitrogen. Second is these proteins, nucleic acid and vitamin, hormones all are composed of this nitrogen. This is most important. Deficiency of nitrogen that's shown in first in senescent leaf. If we see a senescent leaf, the deficiency of exact mineral or new mineral that is known as nitrogen. This nitrogen first shown in a senescent leaf. What is senescent leaf? Senescent leaf is the leaf that dies and remobilize nutrition from one younger cell, so that this, let's consider this is senescence leaf. In senescence leaf, nutrient are remobilized to the other young part, so that these plant or these cells are survive betterly. Next mineral, phosphorus. So what is phosphorus? This phosphorus are absorbed in H2PO4 and HPO4. This is H2PO4 and HPO4. These are constitute the main constitution of cell membrane. We know cell membrane composed of phosphate and lipid. So this is the basic composition of phosphorus. Certain proteins, nucleic acid and nucleotide also consist. We know DNA, in DNA found phosphate, sugar and nitrogen base. So this is phosphate is the backbone of nucleotide. Next concept or next mineral, iron. This iron are absorbed in plant in Fe3 plus. This is called ferric, and that helps is a cofactor of this enzyme known as catalase. This catalase enzyme that helps in during different types of scavenger, like protect cell from different types of oxidative reaction. And second thing, this iron helps in chlorophyll formation and biosynthesis. Means iron helps in synthesis of chlorophyll also. Next, magnesium. So what is the function of magnesium? This magnesium are absorbed in Mg2 plus, and this activates different type of respiration and photosynthesis enzyme that activate different types of respiration and photosynthesis enzyme, helps in synthesis of RNA and DNA. That most important thing is it also helps in synthesis of chlorophyll. As like iron, this is also help in synthesis of chlorophyll and present also in chlorophyll. Next, sulfur. Sulfur is absorbed in SO4 2-. This is called sulfate. Sulfate is the observable form of plant, and this sulfur constitute of different amino acid. In different amino acid we see sulfur. One is called cysteine, methionine. These are composed of sulfur, and different types of vitamin that also composed of sulfur like thiamine, biotin, coenzyme A, and ferredoxin. These enzymes and vitamins and amino acid also composed of sulfur. Next is manganese. What is manganese? Manganese is observed in the form of Mn2 plus, and that helps in photosynthesis, respiration, and nitrogen metabolism. But the main thing in manganese, this is most important, this is called photolysis of water. What is photolysis of water? During photosynthesis, water molecules are breakdown. This breakdown occur by one of the mineral called manganese. Boron. Boron are observed in borate and tetraborate, and these are main function is uptake of calcium, that helps in the uptake of calcium. If the uptake of calcium occur, this calcium helps in different metabolic activity, that helps in different metabolic activity. And the main factor of boron that helps in germination of pollen, and helps in cell differentiation. So what is cell differentiation? Let's consider this is a cell. This cell divide to another cell and finally divide. Let's consider this is a before this is a skin cell, and finally divide to skin tissue and forming a specific skin. As like in plant, let's consider a specific xylem tissue or xylem cell that divided and finally form xylem. Next is that help in carbohydrate translocation. Carbohydrate translocation means that it transfer carbohydrate from one place to another place, that help by boron. Next, zinc. Zinc is observed in Zn2 plus, and that activate one of the enzyme known as carboxylase. Zinc is activate one of the enzyme called carboxylase that helps in synthesis of oxygen. Zinc helps in synthesis of oxygen. This oxygen also known as IAA. This is the most important thing that synthesize oxygen. Next, calcium. This calcium that helps in, or this is observed in Ca2 plus, and this is present mostly that helps in growth of root tip, that helps in growth of root tip. If this is a root tip, that helps the growth. And the composition we found in cell middle lamella, that is called calcium pectate. Calcium pectate is mostly found in middle lamella that regulate different metabolic activity. Next is potassium. So what is the function of potassium? Potassium is balance anion and cation balance. Anion means negative and cation means positive ion balance. This is the function of ion balance. And next, photosynthesis, that helps in photosynthesis also. The major thing of potassium that helps opening and closing of stomata. And another importance of this potassium that helps in giving turgidity or turgor in cell. So what is turgor in cell? Turgor in cell, let's consider this cell observe water and create a pressure on cell wall, and shape is as like, and this condition is known as flaccid. Flaccid means that have loose water, but turgid means that absorb water and create a pressure on cell wall. So this condition is known as turgidity, and it is due to the ion called potassium ion. Next, copper. Copper is absorbed in Cu2 plus and that helps in redox reaction. Oxidation and reduction are known as redox reaction. Next, chlorine. Chlorine are absorbed in Cl minus. The major function of chlorine that help in water splitting reaction in photosynthesis so that oxygen evolve, means oxygen molecule are released due to this splitting of water during photosynthesis. We completed different things. Now I'm discussing the what is the exact application, different exact application of these minerals. So one is known as a dealing of flower. So what is dealing of flower? Dealing of flower, normally we know flowering, let consider a flower that reaches for a proper flowering 2 months. If it delay, if it take 5 months, then flowering is known as delaying of flowering. For this delaying of flowering, this this is called molybdenum, sulfur and nitrogen. This molecular most or these minerals are mostly involved in the delaying of flower. Next is necrosis. So what is necrosis? Necrosis is the premature cell death. This is known as premature cell death. This premature cell death is caused by calcium, copper, potassium and magnesium. And the next concept, inhibition of cell division. We know cell is composed of different spindle fiber and other component. Cytokinase, karyokinase occur in cell division. But if deficient in these minerals, then it causes inhibition of cell division. These minerals are sulfur, molybdenum, potassium and nitrogen. Now bilateral elements. These bilateral elements are selenium, sodium, silicon and cobalt. Critical element. This critical element are most important element for the plant called sodium, potassium and phosphate. Next concept is chlorosis. So what is chlorosis? Chlorosis is the that green color of plant that convert into pale yellow due to the degradation of chloroplast. If the chloroplast degrade in the leaf, green leaf, that cause change in color. Mostly we see in morphologically the color from green to that converted into pale yellow. So for this, that involve different types of nutrient or minerals: sodium, potassium, magnesium, manganese, iron, sulfur, zinc and molybdenum. These are the total application of different mineral and how this mineral affect on different types of disease deficient and how that affect plant growth and development also. Next concept called nitrogen cycle. In nitrogen cycle we see the nitrogen that converted into this nitrogen converted into ammonia, in which we see nitrogen, this nitrogen converted into ammonia. This conversion of ammonia is known as nitrogen fixation, or this is called nitrogen fixation, means that nitrogen convert to ammonia. This ammonia further converted into nitrite and nitrate. This conversion of nitrite to nitrate through this process, this is known as nitrification. In nitrification we see the ammonia, this ammonia that converted into first nitrite, this is NO2 minus and NO3 minus. This NO3 minus nitrate convert nitrate to nitrate, this is called nitrification. Then further this nitrate absorbed by a plant. After observing this plant, that sent to animal, and then after plant and animal both are dead. After death, that is known as decaying of material. This decaying of material further the nitrogen that converted into different form to the different form of nitrogen. This process is known as denitrification. So further I am discussing this thing, nitrogen cycle. In nitrogen cycle we see the first nitrogen converted into ammonia. After conversion of ammonia, that converted into nitrite to nitrate. So first conversion of nitrogen to ammonia is called nitrogen fixation. Then ammonia to nitrite and nitrite to nitrate. This conversion is known as nitrification. Then this nitrate that absorbed by plant, and that plant is eaten by some animals. Both plant and animal are dead. After dead or decaying, this nitrogen compost mix in the soil in the nitrate form, and this nitrate further converted into different form of nitrogen. This process is known as denitrification. So this is the all overall discussion of nitrogen fixation. Next is here we see first this nitrogen convert into ammonia. This ammonia oxidize to form nitrite. This nitrite with hydrogen molecule and water molecule. This conversion, the product is nitrite with hydrogen molecule. This is the hydrogen molecule. And next we see this nitrite further oxidize to convert into nitrate, and this nitrate is absorbed by plant. For the nitrification, we see here for the nitrification an enzyme and bacteria are involved. These bacteria are known as Nitrosomonas, Nitrococcus and Nitrovector. This Nitrosomonas, Nitrococcus and Nitrovector that helps in the nitrification. So what is nitrification? In nitrification we see the conversion of ammonia to nitrite. This nitrite further converted into nitrate, and for this we see Nitrosomonas, Nitrococcus and Nitrovector are involved. Next concept is denitrification. For the denitrification, different bacteria that involve, one is called Pseudomonas, Thiobacillus, Pseudomonas and Thiobacillus. These are involved in denitrification. Next concept, biological nitrogen fixation. So what is biological nitrogen fixation? We see the mostly in biological nitrogen fixation symbiotic relationship. This symbiotic relationship means plant with bacteria that helps in nitrogen fixation. We see the bacteria that mostly found in root cell. This bacteria combinedly helps in the fixation of nitrogen, so that helps in the product absorb nitrogen, and then after this nitrogen converted into different form. So here you see this nitrogen N2 converted into NH3 through a enzyme. This is called nitrogenase. This process is known as biological nitrogen fixation. In biological nitrogen fixation, we see some bacteria. One is called free living bacteria. One is called Weijerichia, Azotobacter and Rhodospirillum. How to remember this thing? Remember through BAR or bar, that is Weijerichia, Azotobacter and Rhodospirillum. These free living bacteria are involved in biological nitrogen fixation. Also symbiotic bacteria, these are called rhizobium and frankia, that helps in symbiotic association with different types of roots. This rhizobium found in legumes plant and frankia that found in non-legumes plant of root. This found in non-legumes plant of root. This is the legumes plant of root. And this is rhizobium is rod shaped. And this frankia, next is frankia that produce nitrogen fixing nodules on the root of ulnus. Mostly one of the root, this is not a legume plant, and this plant name ulnus, this ulnus root combine with frankia and finally shows nitrogen fixation. And some other bacteria like anabena and nonstuck also helps in nitrogen fixing. Another most important concept, this is called nodule formation. So what is nodule? Nodule is a mature nodule growth like structure or tumor like a swelling structure. That swelling structure found in root inside the cell. This swelling structure or nodule that helps in biological nitrogen fixation. So we study how this rhizobium bacteria that helps in biologically functioning or producing nitrogen or fixing nitrogen through enter into the root cell. So here is the root cell. This is the legumes plant. In a legumes plant we see a bacteria, this is called rhizobium. This rhizobium found in here we see this soil, and this is the root hair. At root hair, near root hair, this bacteria are found and secrete some chemicals. These chemicals reaches this root hair. After reaching this root hair, we know this root hair constitute of different type of cells. It consists of different type of cells, and cells have a receptor. This receptor taken some signal from the bacteria. After taking some signal from this bacteria, then after the cells send also some signals to the bacteria, so that after some time this bacteria enter into this root cell, and division of this bacteria on root cell that forming a nodule like structure. This is known as mature nodule like structure. So that this structure is called infection thread containing bacteria. Means we see here infection with a bacteria, but here that form a nodule, a swelling structure with mature nodule. This mature nodule helps in fixation of nitrogen. So this is the overall concept of nodule formation. Next, nitrogen cycle. In nitrogen cycle we also discuss how the things are happen. Here we see the enzyme nitrogenase. This enzyme nitrogenase helps in this N2 to NH3, and this nitrogenase require a cofactor known as molybdenum and iron. This nitrogen enzyme attach first N, this is called N2. First the N2 molecule attached to this binding of substrate, means the enzyme nitrogenase. This is the nitrogenase enzyme that bind to the substrate known as N2, and a reduction occur, so that two molecules of proton are added here, one is here and another is here. Then after some time we see also reduction occur, two molecules of hydrogen and hydrogen here binding. After some time we see one molecule of hydrogen here, one molecule of hydrogen here, hydrogen here, here, means in this side 3 hydrogen, at upper 3 hydrogen. Total 2 molecule of NH3 are formed. How this nitrogenase that converted nitrogen to ammonia. This is the overall discussion of enzyme action on substrate and the conversion of substrate to product through a chemical mechanism. Next concept, nitrogenase. What is nitrogenase? Nitrogenase is known as biological or nitrogen fixing enzyme that helps in convert N2 to NH3, and this enzyme constitute of different proteins. These proteins consist of mineral known as molybdenum and iron. This molybdenum and iron act as a cofactor for the nitrogenase. This enzyme mostly sensitive to the oxygen. Means if the oxygen bind to the nitrogenase, then the nitrogenase enzyme cannot work properly. But if this enzyme not bind, so to protect from this oxygen, it have another system. This is known as leg hemoglobin. This is also called oxygen scavenger. What is scavenger? Scavenger means mask the oxygen or protect from the toxicity of oxygen. Scavenger means one of the rescuer. Another concept, inactivation of nitrogenase by oxygen poisoning is prevented by leg human. This is the exact point. Inactivation of nitrogenase by oxygen poisoning is prevented by leg hemoglobin. Means if we provide leg hemoglobin, then oxygen cannot bind at this active site, so the enzyme work properly. Next concept, nitrogenase in root nodule of legumes plant produce ammonia and hydrogen. The product is ammonia and hydrogen. In root nodules, mostly root nodule of legumes plant. This is the root nodule of legumes plant. We see this nitrogenase enzyme that produce N2 to ammonia and hydrogen. So the product is not only ammonia and hydrogen. So this is the overall discussion of enzyme nitrogenase. Next concept is called conversion of ammonia. Ammonia is toxic for plant, so it required to convert in different form of amino acid or other form. So first I am discussing how this ammonia that converted into plant. NH4 ion is the toxic for plant, and so it converted into amino acid and other molecule also. So first one is called reductive amination. In reductive amination, we see this ammonium ion with alpha ketoglutarate with NADPH that produce glutamic acid. This glutamic acid is or glutamic amino acid. This is a amino acid, and this amino acid formation occurs through enzyme called glutamic dehydrogenase. This is the glutamate dehydrogenase. This glutamate dehydrogenase convert ammonium plus alpha ketoglutarate plus NADPH to glutamic acid. This glutamic acid is called amino acid through an enzyme. This enzyme is known as glutamate dehydrogenase. And next is transamination. So what is transamination? In transamination, we see the transfer of amino group from one molecule to another molecule. So one is called amino donor and amino acceptor. Means one molecule that give amino group and other molecule that accept this amino molecule through a enzyme. This is called transaminase. This enzyme helps in transamination. This is called transaminase, and one molecule present for donating and another molecule for accepting. So this is the all overall